Hello students, in today's session we will discuss structure of polynucleotide chain. In the last session we discussed chemical composition of nucleotide, and you already know that nucleotides are the building blocks of nucleic acids. When you define nucleic acids, they are the macromolecules or they are polymers of nucleotide. Nucleotide are the building blocks of nucleic acid, and each nucleotide is made up of three components: pentose sugar, nitrogen base and phosphate group. Briefly, we will recall the components of nucleotides. The first one is pentose sugar. 5-carbon sugar. In case of DNA, you find deoxyribose sugar. In case of RNA, you find ribose sugar. The second component is nitrogen base. There are two types of nitrogen bases. One is purine, second one is pyrimidines. There are two types of purines. One is adenine, second one is guanine. And there are two types of pyrimidines: cytosine and thymine. The third component is phosphate group, and you already know that nucleic acids are acidic in nature. Why? Because of phosphate group, and phosphate group is a derivative of orthophosphoric acid. When you talk about nucleotide, the first nucleoside is formed. And you already know that when nitrogen base linked to a pentose sugar through a bond called N-glycosidic bond that results in formation of nucleoside. So look at here, in between base, nitrogen base and sugar, a bond formation takes place, and that bond we called as the N-glycosidic bond. And you remember one thing: if the nitrogen base is purine, then 1,9-N-glycosidic bond formation takes place. If it is pyrimidine, 1,1-N-glycosidic bond formation takes place. As a result of this, there are 4 nitrogen bases, and all these nitrogen bases link to respective sugar or pentose sugar to form nucleosides. So 4 types of nucleosides are formed. Soon after the formation of nucleoside, nucleoside combines with the phosphate group or phosphoric acid to form nucleotide. And again, you remember one thing. So in between nucleoside and phosphoric acid, a bond formation takes place, and that bond we called as a phosphoester bond. When phosphate group is linked to 5' hydroxylic group of nucleoside through phosphoester bond to form nucleotide. So phosphoester bond. Again, you already know that there are 4 nucleosides, and all these nucleosides combines with phosphate group to form 4 nucleotide. Let us see. So come back to the nucleoside. 4 types of nucleoside. One is deoxyadenosine, second one is deoxyguanosine, third one is deoxythymidine, and fourth one is deoxycytidine. And all these nucleosides combines with the phosphoric acid or phosphate group to form respective nucleotides. Let us see. Deoxyadenosine combines with phosphate group to form deoxyadenosine monophosphate, deoxyadenosine monophosphate, or it is called deoxyadenalic acid. And deoxyguanosine combines with phosphate group to form deoxyguanosine monophosphate, DGMP, or deoxyguanalic acid. Deoxythymidine combines with phosphate group to form deoxythymidine monophosphate, or DTMP, or deoxythymidalic acid, And last one, the deoxycytidine combines with phosphate group to form deoxycytidine monophosphate, DCMP, or deoxycytidalic acid. So 4 nucleotides are formed. When 2 nucleotides link together to form dinucleotides, when 3 nucleotides link together to form trinucleotides, when many nucleotides link together to form polynucleotide chain. Let us start with the first one. 2 nucleotides are linked through 3', 5' phosphoester linkage. That means, so you remember one thing, so in between nucleoside and phosphate group, a bond formation takes place. For example, this is a nucleoside, pentose sugar and nitrogen base, and this nucleoside linked to the phosphate group through phosphoester linkage, and this is another nucleotide. So in between this nucleotide and this nucleotide, a bond formation takes place in between 3', 5' hydroxylic group of nucleoside through phosphodiester or phosphoester linkage that results in formation of dinucleotide. If 3 nucleotides, 1, 2, 3 nucleotides join together to form trinucleotides, when many nucleotides join together to form polynucleotide chain. This is polynucleotide chain. So when you look at the polynucleotide chain, the polynucleotide chain has at the 5' end of the sugar free phosphate moieties. This is 1, 2, 3, 4, 5. Okay, 5' end of pentose sugar, we find free phosphate moiety. Okay, and it is called 5' end of polynucleotide chain. Okay, at the other end, look at here, at the other end, 1, 2, 3. Okay, the 3' end of pentose sugar has free hydroxylic group. Here you find free hydroxylic group at 3' end of pentose sugar. So look at here. Here in the pentose sugar, 5' end of pentose sugar has phosphate group. At the opposite, okay, 3' end of sugar has free hydroxylic group. Okay, so that's why it is called 3' end of polynucleotide chain. 3' end of polynucleotide chain. Okay, so one end you find 5', the other end you will called as a 3' end of polynucleotide chain. And look at the polynucleotide chain. The backbone of polynucleotide chain is formed by repeated sugar and phosphate. Repeated sugar and phosphate, while the nitrogen bases they projects from the backbone. They projects from the backbone. Is that clear? So this is about the polynucleotide chain. Look at here. So at the end of free phosphate, one end free phosphate moiety, at the 5' end of pentose sugar, which is referred as 5' end of polynucleotide chain, whereas the other end, opposite end, at the other end you find 3' end of pentose sugar has free hydroxyl group, and it is referred as 3' end of polynucleotide chain, and the backbone of polynucleotide chain is formed by due to repeated sugar and phosphate. This much we discussed in the last session. Let us move on to the structure of polynucleotide chain or double helix model of DNA. The double helix model of DNA was proposed by two biologists, James Watson and Francis Crick, in the year 1953. So James Watson and Francis Crick proposed double helix model of DNA. So based on two observations. One is X-ray crystallography. The first observation is X-ray crystallography, and second one is base equivalence rule or observation made by Erwin Chargaff. Let us start with the first one, X-ray crystallography. The two workers, Maurice Wilkins and Rosalind Franklin, they worked on X-ray crystallography or X-ray diffraction data on DNA. Based on the data, they finally came to conclusion that DNA is made up of two polynucleotide chain. The question arises, what is this X-ray crystallography? It is a technique to determine three-dimensional structure of large molecules or macromolecules. So based on X-ray crystallography, the DNA is made up of polynucleotide chain. So based on this data, James Watson and Francis Crick proposed double helix model of DNA. The second observation made by Erwin Chargaff. It is commonly known as the rule of base equivalence. What is this? A rule of base equivalence andre enu? The amount of purine is always equals to the amount of pyrimidine. So amount of purine is always equals to the amount of pyrimidines. That means the amount of adenine is always equals to the amount of thymine. One. So A equals to T or A by T equals to 1. Second one, the amount of cytosine is always equals to the amount of guanine. That is, C equals to G or C by G equals to 1. So that means the sum of purines is equivalent to the sum of pyrimidines. Polynucleotide chain olaga ಎಷ್ಟು purines irtava ಅಷ್ಟೇ pyrimidines irtava in double stranded DNA. Because Erwin Chargaff rule is applicable for only for double stranded DNA. That means in one strand ಎಷ್ಟು purines irtada adra opposite strand enirtada ಅಷ್ಟೇ pyrimidine irtada. That means the sum of purines is always equals to the sum of pyrimidines. That is, A plus G equals to C plus T, or A plus G divided by C plus T equals to 1. So, the ratio between A plus D divided by C plus G, okay, is variable. It is not equal, variable. Sometime in one strand, okay, purines ಮತ್ತು pyrimidines ratio ಏನಾಗ್ತದೆ, variable ಆಗ್ತದೆ. So based on this, James Watson and Francis Crick proposed a double helix model of DNA. Double helix model of DNA. For this, they proposed or put forth three-dimensional structure of DNA. For this, they got Nobel Prize in the year 1962. Along with James Watson and Francis Crick, Maurice Wilkins also shared Nobel Prize in the year 1962. Look at here. When I talk about double helix model of DNA, what are the salient features of double helix or DNA, double helix structure of DNA? The first, the salient feature or characteristic feature of DNA is it is made up of two polynucleotide chains. Two polynucleotide chain where the backbone is constituted by repeated sugar and phosphate. Look at here. This is one polynucleotide chain and this is another polynucleotide chain. So DNA is made up of two polynucleotide chain, and the repeated sugar and phosphate forms the backbone. Okay, backbone. And whereas the nitrogen bases are located right angles to the backbone, that means they are project inside. Nitrogen bases project inside. This is the first characteristic feature. Okay, we will move to the second one. So you already know that the two strands, two polynucleotide chains are anti-parallel polarity. They have anti-parallel polarity. That means the one strand is inverted in relation to the other. When you talk about the structure of DNA, it is compared to the twisted ladder. That means both the strands run parallelly and does not crosses each other. Okay, twisted ladder ಗೆ ನಾವೇನು ಮಾಡ್ತೀವಿ, ಕಂಪೇರ್ ಮಾಡ್ತೀವಿ. ಎರಡು ಸ್ಟ್ರಾಂಡ್ಸ್ ಈಕ್ವಲ್ ಡಿಸ್ಟೆನ್ಸ್ ಲಿಂದ ರನ್ ಆಗ್ತಾವ. ಓಕೆ, ಎರಡು ಸ್ಟ್ರಾಂಡ್ಸ್ ಎಲ್ಲಿ ಓವರ್ಲ್ಯಾಪ್ ಆಗಂಗಿಲ್ಲ. So one strand is inverted in relation to the other. One strand is inverted in relation to the other. That means one strands run, okay, or has 5 prime 3 prime polarity, or one strand runs in 5 prime 3 prime direction. That means here the phosphate moiety is free at 5' end, and in 3 prime end you find free hydroxylic group. Other end 3 hydroxylic group of pentose sugar is free. That's why it's called 3 prime end. So one strand runs in 5 prime 3 prime direction, whereas the other strands run 3 prime 5 prime direction. Okay, 3 prime 5 prime direction. So this is second characteristic feature. The two strands have anti-parallel polarity. That is, one strand or one chain has polarity 5 prime 3 prime direction, whereas the other strand has 3 prime 5 prime polarity. So this is second characteristic feature. And you already know that the backbone is constituted by repeated sugar and phosphate, whereas the bases are projected inside at right angles to the backbone. So this is backbone, right angles to the backbone. The bases, nitrogen bases are projected inside. So these nitrogen bases in the polynucleotide chain, they forms the steps of the twisted ladder. Steps of the twisted ladder, and both the strands are connected to each other with the help of hydrogen bonds. That means the base of one strand is always paired with the base of other strand or opposite strand and third thing. The base of one strand is always paired with the base of other strand. Okay, or the bases of two strands are held together by hydrogen bonds forming the base pairs. Let us, the base pairing due to the formation of hydrogen bonds. Look at here, this is one strand. The base pairing takes place between two strands with the help of hydrogen bonds. Adike naವೇನಂತ ಹೇಳ್ತೀವಿ, base pairing ಅಂತ ಹೇಳ್ತೀವಿ. So this is third character. The bases in the two strands are held together by hydrogen bonds, forms the base pairs, and during base pairing what happens, the adenine, the base pairing takes place in between purines and pyrimidines, or double ring purines and single ring pyrimidine. Because of base pairing, it creates a constant diameter of DNA molecule at 2 nanometer to nanometer. They have a constant diameter because of what? Because of base pairing. Because of base pairing. So during base pairing, adenine base pairs with thymine, adenine base pairs with thymine with the help of two hydrogen bonds. Okay, two hydrogen bonds, and cytosine base pairs with guanine with three hydrogen bonds. And the base pairing always takes place between purines and pyrimidines. Look at here, if the one strand has the adenine, it base pairs with thymine with the help of two hydrogen bonds, and the one strand has cytosine, the other strand cytosine base pairs with guanine with the help of three hydrogen bonds. This is we called as a complementary base pairing. Complementary base pairing, because both the strands are complementary to each other. Okay, so this is a third characteristic feature. We'll move on to the and one more thing, the basis of one strand will determine that of other on the other strand, opposite strand. If the one strand, for example, you will consider this strand. Okay, look at here, if the one strand has adenine, thymine, cytosine, guanine and thymine. Look at here, the other strand, adenine base pairs with thymine, thymine base pairs with adenine, cytosine base pairs with guanine, and guanine base pairs with cytosine, thymine base pairs with adenine. If the base pairs of one strand is known, automatically in DNA, we can easily determine the base pairs of opposite strand. So the other strand will have what? TAGCA. This we called as a complementary base pairing. Complementary base pairing. We'll move to the fourth characteristic feature of DNA, or salient features of DNA. You already know that this is a twisted ladder. Okay, ನೋಡಿ, ಎರಡು ಸ್ಟ್ಯಾಂಡ್ಸ್ ಹೆಂಗಾಗಿದೆ, ಅಪೋಸಿಟ್ ಆಗಿ ಪ್ಯಾರಲಲ್ ಆಗಿ ರನ್ ಆಗ್ಯಾವ, ಎಲ್ಲಿ ಓವರ್ಲ್ಯಾಪ್ ಆಗಿಲ್ಲ. ಇಟ್ ರಿಸೆಂಬಲ್ಸ್ ಲೈಕ್ ಟ್ವಿಸ್ಟೆಡ್ ladder. The two chains are coiled at a right-handed fashion, and the pitch of helix, the pitch of helix is about 3.4 nanometer. That means The one complete turn, the length of one complete turn is about 3.4 nanometers. Okay, so 360 degrees when the turn bರ್ತದೆ, or so distance between two minor grooves, we'll discuss later what is these minor grooves and major grooves. The distance between two minor grooves or major grooves we called as a one complete pitch or one complete turn, and the length of one complete turn is 3.4 nanometer, and one complete turn is made up of 10 base pair. 10 base pair. If it is bDNA or DNA is coiled in a right handed fashion, the pitch of helix is 3.4 nanometer. Okay, and the distance between two successive base pair is 0.34 nanometer. 34 nanometer. So the distance between two successive base pair is approximately 0.34 nanometer. This is fourth characteristic feature. And we'll move on to the last one. The plane of base space. So this is a plane of base space which stacks over the other in the double helix. Which in addition to hydrogen bonds, okay, these base pairs in addition to hydrogen bonds that provides stability to the double helical structure. So these are the 5 characteristic features of or salient features of double helix structure of DNA. I'll repeat once again. The first one, it is made up of 2 polynucleotide chain. Second one, the 2 chains have antiparallel polarity. And third one is the bases of 2 strands are held together by hydrogen bonds, forms the base pair. And the third one is the one complete turn, the length of one complete turn is 3.4 nanometer. And in one complete turn, how many base pairs are present? 10 base pairs are present, and the distance between 2 successive base pair is approximately 0.34 nanometer. And the last one, the base space the plane of base space stacks over the other in the double helix. In addition to hydrogen bonds, they provide stability to the double helical structure. These are the characteristic features of DNA. Next, we'll move on to this is a structure, 3-dimensional structure of DNA. And you find 2 types of grooves. Can you see this? These are deep grooves. Can you see this? Deep grooves, and these are narrow grooves. Or we find 2 types of grooves. One is major grooves and minor grooves. The length of one complete pitch can be calculated by the distance between 2 minor grooves, or the distance between 2 major grooves we call it as a one complete pitch or one complete turn. So the 2 types of grooves are produced in the helical twist on outside. One is major or wide grooves of 2.2 nanometer width. Then other one is narrow grooves or minor grooves of 1.2 nanometer width. So these are the characteristic features of double helix model of DNA. So in the next session, we will discuss the packaging of DNA. Thank you.